Okay, so our final lecture for this course is going to be on tumor immunology. And so, of course, tumors are going to develop um, for many reasons. And you can have pathogens that can induce tumors in lymphoid cells. Um, and it's important to understand that the immune system, of course, can only respond to what they consider non-self antigens. And so tumors are difficult to target by the immune system. However, we will talk mostly about lymphoid tumors and how they can be um, dealt with by your immune system. So with respect to tumors associated with lymphoid malignancy, you have genetics that can play a role, as well as there are some pathogens that can, what we call, transform cells, such as your herpes viruses. And then you develop tumors, and you can develop a immunity to tumors, and this can lead to cancer immunotherapy, in which it is a targeted immunotherapy to try to induce an immune response to target the tumor and eliminate the tumor within a person. So we have lymphoid malignancies that are going to affect adaptive immunity. And so the malignancies are going to be from a single lymphocyte or plasma cell. And so each of the malignant populations are going to have identical immune receptor rearrangements because they arise from a single cell, or they're going to express identical immunoglobulins or release, if they're a plasma cell, you're going to have a lot of the same antibody being released. So these types of tumors in the lymphoid system are going to be monoclonal. So it's one cell gives rise to many. And so they look the same. If it's, for example, I'm just picking on flu, if it's a flu hemagglutinin T cell that becomes malignant, now you're going to have a lot of flu-specific hemagglutinin T cells because they're going to divide, because that's what malignancies do. And so you look for monoclonality within your lymphocyte population in order to help to identify these types of tumors. Now, we do have different types of cells that can cause lymphoid tumors. So we have acute lymphoblastic leukemia, which is ALL, and this is usually going to come from pre-B cells that is rapidly dividing, and this is very aggressive. Sorry. So this is a very aggressive tumor, and untreated, it can kill within weeks of diagnosis. We also have, this is ALL. We have myeloma. Myeloma is going to come from your plasma cells. And so these are mature, slow growing, and they're going to secrete monoclonal antibodies, of course, monoclonal immunoglobulins, of course. So a myeloma patient can survive with years without treatment because it's not very aggressive. And then, of course, we have our T cell malignancies. These are going to be more rare because T cells don't get infected with Epstein-Barr virus. And so Epstein-Barr virus can transform B cells into malignant cells. And then we have chronic lymphocytic leukemia, or CLL. So this is acute. And over here we have chronic. And so this is going to be different than acute because these are mature B cells, and the malignant cells are present in the blood, whereas in your acute lymphocytic leukemia, these are going to be in the bone marrow and the blood because they are pre-B cells. So they will be released because they divide, but they're also going to be found in the bone marrow. Whereas your chronic lymphocytic leukemia are going to be those B cells that are already circulating, so you don't find that in the bone marrow. In a lymphoma, the malignant cells are present in the lymph node, and you can also have these also in other tissues. But the B cells are commonly going to cause leukemia because we have some herpes viruses that can transform these B cells. So we have Epstein-Barr virus. And by transforming, that means to make them able to survive, make them not able to be killed. I'm blanking on the term I want to use, but I think you understand that. Okay, so our lymphoid malignancies we have a lot of knowledge about them because they grow like crazy, so we can study them. And it's usually a oncogene, a gain of function mutation of a proto-oncogene. And most often it's by translocations. So basically, you're going to move an oncogene and put it, for example, in B cell lymphomas, you're going to move an oncogene and put it into the region where you have antibody genes. So when the antibody is being made, it's also making the oncogene. And then that increase in the oncogene is now going to cause transformation of the cell into a malignant cell. So here are a list of the different types of leukemias. We have our acute lymphoblastic leukemia, and again, those are immature cells. Chronic leukemia are mature B cells, so it's different. Chronic are often associated with EBV or another infection, whereas your ALL are very rare, but it's very aggressive and very deadly. Multiple myeloma are going to be our plasma cells, so this is a very slow, progressive type of disease. And this is where you look for monoclonal immunoglobulins in the blood. You can also see light chain increase in the urine. They're making too much and it's breaking down. T-cell malignancies are more rare. And although human T viruses are thought to maybe be a cause, there's not been yet identified case of that. And so again, leukemia is going to be blood involved, and lymphoma is solid tissue. So when you have a B-cell lymphoma, that means it's in a tissue. So it's in a lymph node, for example. When you have a B-cell leukemia, that means it's circulating in the blood. Okay, so causes of oncogenesis are going to be primarily through chromosome translocations. So when you do recombination in your B cells and T cells as they are developing, those receptor gene recombination events, you're going to have breaks in the chromosomes that might not be repaired properly. And B cell chromosome breaks can also occur when they're undergoing class switching. So segments of your different chromosomes are going to be brought together, and this can commonly move. Well, when it happens and creates cancer, this is where you get an oncogene moved into the region that is highly expressed, such as your antibody segments. So this often is going to have consequences for the lymphocyte. So for example, it might be a lethal translocation, but sometimes these translocations are going to increase cell survival and lead to malignancies. So chromosome translocations can be one lethal to lymphocyte or can also have a positive effect on survival. Now, and just keep in mind that these immune cells are very active. So when you bring a proto-oncogene into a region that is highly expressed in these genes, that proto-oncogene will automatically get expressed as well. And this can lead to permanent activation of the oncogene, which then will lead to cancer. So we have some lymphomas in which we have our MYC gene. And so this is on chromosome 8. And this is, we also have a BCL2 gene, which, BCL2 is the proto-oncogene because it causes or stabilizes your mitochondria and doesn't allow for apoptosis. And this one's going to be on chromosome 18. And what's often seen in our B-cell lymphomas is that you have a translocation of either your MYC or the BCL2 to the immunoglobulin heavy chain gene on chromosome 14. So translocated. And so this will activate or will turn on these two proto-oncogenes. And MYC is going to stimulate lymphocyte proliferation. And this is always going to be in balance with apoptosis, but an overactivated BCL2 protein will protect against apoptosis. And so you get just proliferation of these lymphocytes that have these translocations. So you also have to have, you would think that this would happen more often than it does, and these translocations probably do, but it doesn't always lead to cancer. And so another thing that has to happen are epigenetics. And these epigenetic changes are under massive investigation. For example, methylation of tumor suppressor genes is going to be needed to decrease their function. So remember, it's always a balance between tumor suppression and your proto-oncogenes. And it's the whole two-hit hypothesis. So even though you have a gain of function of a proto-oncogene, you need to have a loss of function of a tumor suppressor gene. So that still kind of holds. Now, one thing to keep in mind is these translocations are increased upon exposure to certain environmental influences such as radiation. So the Hiroshima patients with after the atomic bombs, myeloma was very common. Again, it's a very chronic disease, and it develops over time. But because of exposure of the radiation, caused translocations in individuals. Now, with respect to pathogens, we have our Epstein-Barr virus or herpes. And Epstein-Barr virus is going to be the cause of infectious mononucleosis. It also is a cause of lymphoma and nasopharyngeal carcinoma. Now, EBV does not destroy cells. EBV likes to live inside of cells, especially B cells and memory B cells. And we know memory B cells have long lifespans. And so that means EBV can potentially infect a person for their entire life. And EBV is a large virus. It's a DNA virus. So it has a large genome, and it's going to immortalize the cell. Now, this is going to drive proteins that cause the B cell to proliferate. It also is going to turn on genes that inhibit apoptosis. EBV is a herpes virus, so herpes viruses naturally prevent MHC class 1 upregulation, expression, so it's going to downregulate MHC class 1 too. So EBV is going to influence lymphomas. These are tumors that form inside of tissues by the immune system, and they're going to be B cell lymphomas. You're going to have both Hodgkin and non-Hodgkin lymphomas influenced by EBV infections. Now, EBV is going to also cause or influence a lymphoma called Burkitt lymphoma. And you have polyclonal activation of B cells by both malaria and EBV in regions of the world where malaria is endemic. And this helps to promote this type of lymphoma. You also are going to have the potential for enhanced translocations just simply because EBV causes your immortalization of the cell and cell division. So just by increasing the replication of the cells increases the chance that those proto-oncogenes we talked about, BCL2, MYC, are going to be turned or activated permanently by moving them into the area of the antibody genes. So EBV gets into your cells, is able to hide in your memory cells. It's going to be a driver of B cell growth. It's going to be a driver of B cell proliferation. And it's going to lead to a prolonged mononucleosis-like illness, which you are tired, you have just general malaise. And it's also going to be associated with lymphadenopathy. Again, you're going to have your translocations of MYC gene for like Burkitt lymphoma, which promotes the growth of the malignant B cell population. So again, this is going to be a monoclonal type of cancer. You can also have infection with another herpes virus called herpes virus 8, human herpes virus 8. Let me draw this. So this is human herpes virus 8. And human herpes virus 8 is going to cause Kaposi sarcoma in immunodeficient individuals. So typically Kaposi sarcoma was identified with people who had HIV and were progressing to AIDS. Helicobacter pylori is another organism that can lead to a lymphoma. H. pylori is a bacterium, can cause lymphoma. It triggers chronic inflammation in the stomach. It's commonly the cause of stomach ulcers, and it can cause inflammation. And this is going to be a lymphoma in the mucus-associated lymphoid tissues. Treatment of the H. pylori infection can lead to the lymphoma going into remission, so that's important. Okay, so we have, these are the three big ones that your book also talks about are human T lymphotropic virus 1, HTLV1. Um, However, HTLV1 is very rare, and this is going to be a T cell lymphotropic virus. So it's going to cause a T cell malignancy, which is very, very rare. Okay, so as we know, we're going to have a two-hit hypothesis or two-hit events affecting transformation of your cells into cancerous cells. And so there's always going to be a sequence of events. And in this picture shown in your book, it's talking about Epstein-Barr virus infection. And then you have a patient who gets a renal transplant. And prior to a renal transplant, the woman had memory T cells that respond to EBV. Now, after the transplant, you're going to have an immunosuppressive regimen, and it's going to prevent those memory cells from responding to EBV. And so you're going to get an increase in EBV infected cells. So these are these little yellow guys, or EBV. So then you're going to have the outgrowth of these B cells and with the EBV, and this is going to have increased risk for the translocation of proto-oncogenes moving to the area where you have a lot of expression of your antibodies. And so in this case, you're going to get a MYC translocation, and this allows for an increase in proliferation of one of those particular B cell clones that are infected with EBV. And this leads to a monoclonal B cell proliferation. And this B cell proliferation continues even if you take away the immune suppressive drugs. Now, what is happening here is you have all these different events. You have the initial EBV infection is controlled, and then you have an uncontrolled EBV infection, which drives B cell proliferation, because that's what EBV does. And then you gain a MYC translocation, so this is a gain of function of a proto-oncogene, and then that is going to lead or move this towards the development of a lymphoma. And so you can see that over time this developed. Okay, so diagnosis, you can look for abnormal cells, so you can look for the appearance in the blood of cells. In the presence of acute leukemia, for example, and again, that is malignant cells circulating in the blood. The presence of very high immature cells or blasting cells is a good diagnosis. We can also use now flow cytometry. So this is a blood smear example, and then over here is flow cytometry. So flow cytometry is looking for B cells, and those are CD19 positive cells, and they're using the marker CD5 for T cells. And then in a person who has a malignancy, you can see a population of abnormal cells, because typically T cells do not express a B cell marker, and B cells do not express a T cell marker. But in this case, you have a large population that is expressing both CD19 and CD5, and so that is unusual. The flow cytometry is going to distinguish between acute myeloid leukemia, which is AML, which is caused by neutrophil precursors, from acute lymphocytic leukemia caused by B cell precursors, because NK cells are not natural killers, are neutrophils, and B cell precursors have different markers on their surfaces. Flow cytometry is important also because you can look at many markers. So you can look at a B cell and see, for example, that is expressing a T cell marker. And so they can look at cells at the molecular level and see that they are, in fact, different. Okay, so in this case, we're looking at a myeloma. So you can see with a patient, this was taken from a patient who has bone pain, and again, fatigue, very, general symptoms. Um, you also have, they took a serum sample or a urine sample and found that they had a lot of kappa light chains present in the urine, and this is called the Bence-Jones protein. Okay, so this means that they're making a lot of antibody and it's breaking down, and you can measure the pieces of the antibodies in the urine. You're also going to have a series, a radiograph shows a series of bone lesions, and the bone marrow aspirate is going to confirm that you have an excess number of plasma cells. So for the electrophoresis from the left is going to be the antibodies. So this is the gamma band from a normal. And then you can see a band from a person with a lymphoma or cancer or myeloma cancer. And this has a monoclonal antibody band, which means that you have an outgrowth of some plasma cell that's making the same antibody. And so this is going to be indicative of a plasma cell that has gone cancerous. Now the skull is again showing hypercalcemia due to increased bone reabsorption, and which contributes to the patients having renal failure. And then of course you take a bone marrow aspirate, and I should have showed a normal picture of a bone marrow aspirate because a bone marrow typically looks very different. You have neutrophils, red blood cells, you have lymphocytes, you have macrophages, you have eosinophils, you have just, it looks like a smorgasbord of cells. And so when you take a bone marrow aspirate and it looks uniform, that is not good. And in this case, it's showing an increase of plasma cells. And so this is again, confirming a myeloma type of diagnosis. And so we also have immunity to tumors. So the adaptive immune response, again, is always going to respond to something it sees as being different. You can have tumor cells turn on genes that cause expression of proteins that are not normally present in a particular tissue or at a particular stage of development. And so you're going to have things called tumor-specific antigens. So tumor antigens. Now, tumor antigens are going to be, again, expressed by the tumor, and they can potentially be recognized by the immune response. So you can have developmental proteins that are normally expressed during development, and then they get turned off. And tumor cells, because they are cancerous and they're growing out of control, sometimes can re-express these developmental proteins. And so there's an example of carcinoembryonic antigen, or CEA. And this is normally expressed during fetal life and then gets absent on adult life cells, but it can be abnormally expressed in gastrointestinal cancer. You also have something called lineage-specific proteins, which are expressed in cancers and in the normal tissues from which they arise. So melanoma is a skin cancer of melanocytes. So normal melanocytes and melanoma cells will express the enzyme tyrosinase, and this is not normally expressed in other cells. So tyrosinase is a specific protein made by melanocytes. So this is how you can identify where the tumor arose from. So when you have malignancy, the tumor obviously left the site where it was the tumor initiated, and it goes to other parts of the body. And you can take a biopsy of tumors, look at what type of proteins they're producing, and if you see tyrosinase, you can say, oh, this derived from a melanocyte. And so at least you know the origin of the type of the cancer. Tumors can also express viral proteins. So we talked about EBV, so Epstein-Barr virus. Human papillomavirus is a cause of cervical cancer, and so the cells in the cervix can express human papillomavirus. And then, of course, we have proteins that are produced during translocations. We talked about our B cell receptor, and our ABL fusion protein. And the product is a translocation similar to a BCL2, MYC. ABL is another proto-oncogene. Now, the developmental proteins and lineage-specific proteins are going to be weakly immunogenic because they were expressed on normal tissues. And so central tolerance. So these are weak targets due to central tolerance. And so the prevention of autoimmunity is important, of course. However, elimination of tumors arising from self-tissue is also important. But our central tolerance favors prevention of autoimmunity, and so it will delete the T cells that are specific for developmental proteins as well as lineage-specific proteins. So that CEA, for example, that is found on colon cancer cells, it does not induce a strong immune response. And then viral infusion proteins tend to be more immunogenic because they do not eliminate T cells through central tolerance specific for them. So fusion proteins look different, similar to when we're talking about type 2 hypersensitivity with anemia, where you get penicillin binding to the receptor of a red blood cell, and now it looks different. Now your immune system can see it. Well, the fusion proteins are very similar. Now they look different, and so potentially the immune system could see them. Vaccination has been used as a method to prevent 2 cancers. So we have vaccination to prevent liver cancer caused by hepatitis B virus. Oops. And then also vaccines, of course, to human papillomavirus reduces the cervical cancer. Okay, so there is evidence that, our immune system can respond to cancers. And the immune system can attempt to eradicate some types of tumors, such as those that express viral antigens. A lot of research in cancer immunotherapy is focused on activating T cells, specific for tumor antigens, such as the tyrosinase that I mentioned earlier in melanoma. Now, that makes sense because T cells can do cell-to-cell killing, so if you can get a T cell activated that will recognize the tyrosinase, they could potentially go and kill all of the cells that are expressing that. But in doing this, there is also a lot of side effects associated with T cell therapy for cancers. So the immune system can damage the normal tissue. Some types of therapies can lead to autoimmunity, etc. Now, the other side is that you have your tumors. So tumors are going to look like our own cells, so they have an advantage there. So they're sort of camouflaged in a way. They are rarely going to induce inflammation on their own, so they're not going to activate so they do not activate the innate response. And via those pathogen recognition receptors. So they don't activate toll-like receptors, etc. This means that the innate immune response usually doesn't even know the tumor is there. And the macrophages that are tissue resident don't let the adaptive immune system know that there is something going on in a particular tissue. And you don't really respond to the tumor growing. Some tumors also decrease MHC. And this can lead to NK cell killing. But NK cells are not as potent as CD8 T cells because there's not as many of them. And so the NK cells might be limited. And you can think about tumors developing as a selective advantage. So you might have one cell that expresses plenty of MHC, and you might have another cell that expresses little. And it depends on which one survives as to their daughter cells, as which one outgrows the other. And that is the case for all of these mutations these tumors are going through. They're just trying to have a selective advantage in order to survive in whatever tissue they're in. So tumors can selectively evade the immune system in other ways as well. Your tumors can create Th2 environments. Some tumors are able to secrete cytokines. They turn on cytokine genes. And of course, the ones that promote a Th2 environment would promote survival of the tumor. And those, of course, that maybe turn on Th1 type of genes might not survive and get outcompeted by the Th2 driving tumors. And so there's a lot of different things tumors do, but one of the big things is it looks like self. So we have different approaches to treating tumors. You have immunotherapy, and so immunotherapy is basically an attempt at making your immune system attack and eradicate tumors that exist inside of someone's body. And again, many approaches have been tried. Most are not successful, and there are a few approaches that have some success. So there are two main categories of immunotherapies. You have passive immunotherapy, and this is where you're going to give an immune response similar to like passive antibodies. And this is the immunotherapies that are passive are in fact usually what's called a monoclonal antibody. You can also have an active therapy in which you're trying to trigger the patient's immune response to respond and kill the tumor. And active actually would be better because it's going to provide lifelong responses to the tumor antigens that you're trying to get it to target. Passive immunity, just like passive antibody therapies for infectious diseases, is going to be short term unless you have to give it continuously. So for passive monoclonal antibody therapies is you can generate in the lab antibodies that can bind with different specificities to tumors. And then because they're antibodies, they have those FC regions that can activate the immune system, complement, get inflammation, etc. And the molecular antibodies can bind to those FC receptors on natural killer cells and lead to antibody-dependent cellular cytotoxicity. So passive are monoclonal antibodies. I'll put lab-derived. And then these antibodies are injected and they bind to whatever antigen is on the surface that they're specific for. And these can be tagged with a toxin, for example. And so this is engineered to deliver a toxin to the tumor cell. And it's specific to the tumor cell because it's binding to that specific tumor antigen. It can activate complement through its FC region. It can bind to a natural killer cell through the FC, and this will undergo apoptosis. And again, the antibodies, monoclonal antibodies, are derived in the lab, and it's usually patient-specific therapy. So these are very tailored to the type of or the type of tumor a patient has. You also have some tumors that have antigens that are going to be involved in signaling pathways, and so you can take advantage of this. There's a molecular antibody that binds to CD20, which is, of course, important during B cell activation. And the molecular antibody binds to CD20, and it can trigger apoptosis in the malignant cell. So a monoclonal antibody that targets CD20. So this is, of course, on a B cell, so B cell lymphoma, for example, and triggers apoptosis. There's also an antibody against HER2. This is the epidermal growth factor receptor on cells of about 30% of all breast cancers. Oops, sorry. And so the antibodies are an antagonist for this receptor and prevent tumor growth. And this one is molecular monoclonal antibody. And so those are just a few examples of this. There are other growth factors that are very generally used by cancer. Again, cancers like to grow, so they are going to use our normal growth factors for this process. So vascular endothelial growth factor is going to be used to stimulate blood supply. And so angiogenesis in a tumor is very important for the tumor to maintain oxygen levels. So monoclonal antibodies that block vascular endothelial growth factor so you can have a monoclonal antibody that targets the growth factor or the receptor. And then that will decrease the ability of the tumor to have vasculature within it. And so that's going to be important to treat colon, lung, and some breast cancers. And this is called B. Now, active cancer therapies are going to have to overcome those central tolerance mechanisms that we know of. And if you can get toll-like receptor engagement by the innate immune system, it can help overcome this tolerance. So there are a lot of cells that are going to be secreting pro-inflammatory cytokines in response to toll-like receptor stimulation. For example, you can use tuberculosis vaccine, that's that BCG vaccine. The tuberculin antigen is very potent at triggering a toll-like receptor. So you can give BCG vaccine. And this is going to trigger toll-like receptor 2 and 4. And this is going to, again, just get the inflammation going. We also have a synthetic ligand for TLR7. And again, this is going to activate your innate response to get your acquired response going. And if your innate response is geared up, again, overcoming that, maybe that tolerance level will be decreased, or the threshold to overcome it will be decreased. And so these are used in some skin cancer and basal cell cancers, for example. And then the BCG can be used for bladder cancers, for example. So in general, you're going to have your tumor cells that are going to be expressing some tumor antigen with MHC class 1. You have a tumor infiltrating lymphocyte, but it's not being activated, even though it does see the tumor antigen. And this is because it has something called CTLA4, which is going to suppress the T cell response. And then over here, you have a toll-like receptor stimulated a cytokine secretion. And in this case, you're promoting inflammation using the BCG or other toll-like receptor type of agonists. And then your CTLA4, in this case, because it's pro-inflammatory, gets switched off, and CD28, the co-stimulatory molecule, now can engage CD80. And then you have an increase in tumor infiltrating lymphocytes. Now this goes back to CTLA4 versus CD28. And CTLA4 binds CD80, CD28 binds CD80. This has a higher affinity. This has a lower affinity. So if they are equal 1-to-1, then your CTLA4 negative signaling, so negative signaling, and this one will be positive activation. So again, if you have 1-to-1, CTLA4 will always win over CD28. However, during inflammation, CTLA4 levels will decrease. CD28 levels now can overcome the break that CTLA4 had and can engage CD80 and become activated. Okay, so here's an overview of immunotherapies. We have our passive therapies and our active therapies. Passive are going to be monoclonal antibodies. Active are going to be ways to activate the innate immune response through ligands that bind to toll-like receptors. We're going to have some that are going to allow for co-stimulation to occur. and then others are going to target T cells. So passive, we're going to have our different antibodies that are going to either be for a lymphoma, breast cancer, or a combination of them. We also have toll-like receptors that are going to engage toll-like receptors, BCGs, used for bladder cancer. We have a basal cell cancer as well as melanoma. Now for T cells, we have therapies that I didn't really talk too much about. We have tumor-infiltrating lymphocytes and engineered T cell receptors. So the engineered T cell receptors are using the CRISPR technology. And there's a lot of things going on in this, but again, it's going to be tailored to a specific cancer. But they're basically able to one approach is you take an antibody, you generate a monoclonal antibody in the lab, and then you take a T cell and you cause that T cell to make or have on its surface this monoclonal antibody. So the T cell still has a T cell receptor and still has all that other stuff, but you're tethering a monoclonal antibody to the surface of a T cell, and then you release that T cell into the body of the patient, and that monoclonal antibody functions similar to the passive antibody. It goes and binds to the specific antigen on the cancer cell, but instead of activating complement and all of those things, it's activating the T cell. And now the T cell didn't do a one-to-one killing, kill that tumor cell, and move to the next cell, and the monoclonal antibody can bind to the next cell, and it sort of bypasses the T cell receptor need in order to kill the tumor cells. So there's a lot of things going on with respect to T cell engineering and cancer. Okay, so that is the end of this. Thank you.